everyone, welcome back. In this video, I will discuss the half-life for 0, 1st, and 2nd order kinetics. So the half-life is defined as the time it takes for the concentration of the reactant to fall 1/2 of its initial value. So it's the time it takes for the concentration of a reactant to fall 1/2 of its initial value. All right, so you can see here in this graph we have concentration versus time, and that this was the initial concentration of our species, and then when half of it's disappeared, it's taking 100 seconds for the sample. After twice of it is disappeared, so 2 half-lives, 200 seconds have elapsed, and then after 3 half-lives, it has taken 300 seconds. And I just want to make note that this is only for a first order reaction, and that's unique to first order, that the half-life is constant. So for this one here, we saw every 100 seconds was the half-life. That's not always the case. That only occurs for first order kinetics, and that for first order kinetics is independent of concentration. So let's go ahead and discuss first order kinetics. So remember from the previous video, I taught you the integrated rate law. And the integrated rate law for first order kinetics was the natural log of the concentration of the reactant at a specific point in time over the concentration of initially is equal to negative kT. Now, if we're talking about half-life, we're saying that the concentration of our reactant after the half-life is equal to half the concentration initially. Knowing this information, we can substitute this here into our integrated rate law to figure out what the half-life would be equal to. So let's go ahead and do that derivation together. So now I have natural log of the 1/2 of the initial concentration over the initial concentration is equal to negative k times the half-life. We're substituting time for the half-life. And as you can see here, the initial concentration cancels out, and so that's why the first order kinetics is independent of that initial concentration. You can see that derivation right there. So now we have the natural log of 1/2 is equal to negative k times the concentration, or not, times the half-life, my apologies. And therefore half-life is equal, and the natural log of 1/2 is 0.693 over k. All right. Okay, so let's look at second order kinetics. So second order kinetics, we have, I'm going to already substitute 1/2 the initial concentration for the concentration of A at a specific point in time into its integrated rate law. So this is what it would look like for second order kinetics. It's equal to K times the half-life plus 1 over the initial concentration of the reactant. And so second order kinetics is dependent on concentration. Nothing cancels out there, but let's go ahead and simplify so that we can isolate the half-life. So I'm just rearranging the equation. Simplifying it some more. And solving for the half-life. So the half-life for second order kinetics is 1 over K times the concentration initially. So definitely dependent on the initial concentration. And then zero order kinetics, just plugging 1/2 the initial concentration of the reactant into the concentration of the reactant at a specific point in time into the integrated rate law. It's equal to negative K T 1/2 plus the concentration initially. And when we rearrange the equation, we get this final answer. Remember, for if you're taking my course, that I provide you that chart that has all the kinetics information you will need, such as integrated rate laws, even shows you the linear plots for zero, first, and second order kinetics. It also shows you the formulas for the half-lives. So you don't need to memorize these formulas. I'm just showing you how they're derived, but you will be provided that information on your assessments. Now we're going to work a problem together that utilizes carbon dating, but I wanted to show you a video first before we work this question together. So let's watch it.
Hey there, and welcome to Brain Stuff. I'm Josh Clark, and this is the Brain Stuff where I explain to you how carbon-14 dating works. Carbon-14 dating, which we also just call carbon dating, is a form of radiometric dating. And all it is measuring the decay of a certain type of atom found in a once living organism to determine when it was last alive. And all of this starts in the upper atmosphere of Earth, where it's constantly bombarded by cosmic rays. And one of the things these cosmic rays do is knock neutrons off of some atoms and protons off of others. And before you know it, the nice, stable, family man nitrogen-14 atom is all wrapped up and gone crazy and becomes what's known as a carbon-14 atom, which is radioactive. Now, carbon-14 atoms aren't the only ones in the upper atmosphere. There's also carbon-12 atoms. Carbon-12 atoms are in much more abundance, and they're pretty stable. Carbon-14 atoms are, again, radioactive and unstable, but they're formed at a reliable, steady rate. So at any point in time, we have a pretty good idea of the ratio of carbon-12 atoms to carbon-14 atoms. Got that? It's important. Now, carbon dioxide is essential to life here on Earth. Plants breathe it in, animals eat the plants, we eat the animals and the plants, and these carbon molecules, carbon-14 and 12, that make up the carbon dioxide, get in everything. What's neat is that the ratio of carbon-12 to carbon-14 found in all these living things here on Earth is pretty much the same as what's in the atmosphere, which means it's predictable. And some very, very smart scientists have figured out that carbon-14 actually decays at a predictable rate, right? Carbon-14, like all radioactive particles, has what's called a half-life. Now, the half-life is the amount of time it takes for the number of radioactive particles in a sample to decrease by half. Carbon-14 has a half-life of 5,730 years. That means that after 5,730 years, the amount of carbon-14 atoms in a, say, plant that you found fossilized will be half of what it was the last time that plant took, well, a breath of life. After 11,460 years, which is 2 half-lives, there'll be a quarter the amount of carbon-14 that was originally present. And then after 17,190 years, you'll have just an eighth of the number of carbon-14, and so on until there's none left. And this is actually one of the limitations of carbon-14 dating, that eventually you're going to go far enough back in time that all of the carbon-14 atoms have decayed. And you don't know whether this took place a day before or 100,000 years before, which means the time limit that you can date things using carbon-14 is roughly 50,000 years. But as long as you are trying to date something that lived on Earth within the last 50,000 years, you can figure out roughly when it was last alive. And the way that you do that is by measuring the rate of decay of carbon-14 atoms compared to the slow and steady carbon-12 atoms that are also present in there. Presto, change out, you've got some carbon-14 dating, and all of a sudden you say, oh my God, this wooden axe handle is 12,000 years old. Pretty sweet stuff. And this whole thing gets even more interesting when you realize that future archaeologists are going to have a lot of trouble using carbon-14 dating thanks to us, humans of the present time. Our industrial activities have been pumping CO2 into the atmosphere and really messing with the ratio of carbon-12 to carbon-14. And even more astounding, all of the nuclear bombs that we set off in the mid-20th century, well, those messed with the atmospheric ratios too. So good luck with all of that, future anthropologists and archaeologists. Sorry.
It's Jake from State Farm.
Sounds like
a really good deal.
Jake. So, that's pretty interesting to hear how that all works. And for my future anthropologists out there, maybe you'll do some carbon dating. But yes, so let's go ahead and work a problem together. So the half-life of the radioactive decay, we're focusing on carbon-14, as he just said, is 5,730 years. And it is independent of initial concentration. So that's a big clue we want to point out. So it's independent of initial concentration. Remember I said the only kinetics that does that is first order kinetics. How long does it take for 25% of the carbon-14 atoms in a sample of carbon-14 to decay? So let's go write down what we are given. So we are given that we have a half-life of 5,730 years. So remember how I said a lot of times the reactions we work with are in seconds, but if we're talking about carbon dating, we're definitely working in years. But it's independent of initial concentration, so something to also write down is first order kinetics, because we will be using the rate law, the integrated rate law for first order kinetics. So something else to note is that radioactive isotope decay is first order kinetics. So just really good clue there. So we'll have to use that integrated rate law. Now I'm only going to focus on the first question just right now. We need to find how long it takes. So we need the time in years, since we're working with a very slow process, for the carbon-14 to decay 25%. Now, if it decays 25%, then what you have is 75% left over of the initial concentration. So make sure that you're looking at the vocabulary that's being used when you're reading the problems. It's saying how long does it take for 25% of those initial carbon-14 atoms in a sample to decay. So if we lose 25%, we're left with 75% of the sample, and I already converted that to the fraction 0.75. So the sample, we want to figure out what time is when we have 75% of the sample left, because 25% of the initial carbon-14 already decayed. So we're going to first calculate what the rate constant is for this process. And we just learned that half-life for first order kinetics is 0.693 over K. And that is equal to 5,730 years. And therefore K is equal to 1.2094 times 10 to the negative fourth inverse years. So going to highlight that because we'll need it in a second. There we go. All right, so then we're going to use our integrated rate law because remember that tells us the concentration as a function of time. I'm going to use the rearranged. So concentration of carbon-14 over the initial. And if we rearrange it to solve for T, that's what we get. Now, if our sample is 75% of our initial concentration, then if you assumed you have a 100-gram sample, how much is left? Good, 75 grams. So you could say the natural log of 75 over 100 times 1 over K and I get the time it takes to be 2,378 years, and that is 2,400 years if we round to two significant figures, like so. And you should always ask yourself if this makes sense. We learned that for half of the sample to decay, it would take 5,730 years. So if only 25% of that decays, it should be approximately half, so about 2,400 years. All right, let's look at the last question together. If the sample initially contains 1.5 millimoles, and it's okay to work in moles here, it's still an amount, how many millimoles are left after 2,255 years? So once again, we will have to use the integrated rate law. And we will use K as well since we're talking about the same process here. So I'm going to write down the integrated rate law. So natural log of C14 at a specific point in time is equal, remember, to negative KT, so negative 1.2094 times 10 to the negative 4th times the time plus the natural log of 1.5. I get the natural log of the C14 sample is equal to 0.1327454. Anytime we want to get rid of natural log, we put it to the exponent here. That helps to cancel that out. So C14 at that particular point in time is 1.14 millimoles, which rounded to two significant figures is 1.1 millimoles. That's left over after 2,255 years. So let's go ahead and review. When we're talking about radioactive decay, we're looking at first order kinetics, but the clue that says independent of initial concentration also told us first order kinetics was taking place. The half-life was provided, which allowed us to figure out what the rate constant is so that we can use it for the remainder of the problem. We also wanted to figure out the time it took for 75% of the sample to be left over or for 25% to have decayed, so to loss. And we used the integrated rate law. You could use either one, it doesn't matter. I wanted to use this one mainly because I wanted to use that ratio. If I have a 100-gram sample, then 75 grams is left, or if you have moles, then that many moles is left. It's just a ratio. When we figured out it took 2,400 years for the sample to decay by 25%. Then we looked at a different scenario, and we knew how much we had initially. We wanted to figure out how much was left over after 2,255 years. Once again, when you're looking at concentration or amount as a function of time, pull out your integrated rate law for that first order kinetics in this case. And then I plugged everything in that I knew, and I wanted to isolate how much of that sample is left over after 2,255 years. And we figure out 1.1 millimoles is still left over from the initial 1.5 millimoles. All right, thank you for watching and see you next time.